All right. Well, hello, folks. Hope you're doing well today. What we're going to do is break down chapter 24 into a few short videos looking at how new species are created. So, as an extension of our discussion of evolution, we're going to look at what events unfold in order to allow life to change to such a point that new species are created. Now, up to this point, we focused mostly on evolution at a smaller scale. It's referred to as microevolution, where we track changes in allele frequency. That's what like the Grants did with their experiments looking at the finches in the populations on the Galapagos Islands. We can look at allele frequency changes pretty readily in populations, and there's extensive research looking into that. But what that does is it tracks the frequency of alleles of populations to see how those allele frequencies change in a population over time. But we know that on grander time scales, life can change to a much greater degree. With macroevolution, you have whole new branches on the tree of life emerge. That happened 500 million years ago with the Cambrian explosion, where the different phyla of animals, like the different types of animals that we see today and that you would study in a zoology class, that's when they all emerged at around the same time. So you can get these big branches that occur. But what we're going to focus on today in chapter 24 is how you get like an individual branch, how you get a single species that crops up. Now, when trying to figure out how speciation occurs, what we need to do is first think about how we're going to define species. There are a few different ways to define species, and I would say arguably the best way is what's referred to as the biological species concept. With the biological species concept, the idea is that members of populations naturally would interbreed with one another and produce fertile offspring. This is the gold standard for defining species because it allows new generations to be created. So, if populations can interbreed and produce fertile offspring, then from a biological standard, they're members of the same species. If populations don't naturally mate with one another or don't produce fertile offspring, if sort of forced to, then they would be biologically distinct species. So the best way, the best option for defining species is this biological species concept. Now, there are other ways of defining species. This was done extensively, especially before there was DNA analysis that allowed you to compare DNA between species. It's the morphological species concept. With the morphological species concept, morph means form. So it's, how do they look? Do they share certain features with one another? And with the morphological species definition, we sort of cluster relatedness based on how organisms or populations look and what features they share or do not share. This is something that happened for a long time in biology with taxonomy, where populations would be sort of compared and put on these trees, looking at evolutionary relatedness, but it was all based on morphology or physical characteristics. Now, obviously, a shortcoming with this is the idea of convergent evolution. With analogous structures, species can converge upon similar adaptations, so while they might not be closely related, they might share certain physical features. So taxonomists could mistakenly think that they're more closely related. Again, think of like sugar gliders and flying squirrels. They've independently evolved these adaptations, but they're not particularly closely related to one another. So the morphological species concept has its shortcomings. Let's see. Oops. Another species definition is the paleontological species concept. And this is looking at like fossilized remains. The trick with the biological species definition is that like more than 99% of all species that have ever existed are extinct. So if there are records of species that once existed in looking at fossils, what we can do is sort of look at features that remain. Now again, this is challenging because, well, remains from fossils can be sort of limited. The image you see there is Lucy. She's probably the most famous Australopithecine. This is sort of an ancient relative to modern humans. And if you look in the image in the middle, these are the fossilized remains of Lucy. So they've got a model created on the left, but the actual parts that they found are in the middle. So if this is a well-preserved specimen, you see that they're just sort of dealing with clues when trying to figure out features. So there might be some distinct features that show sort of like upright posture, or molars give clues to diet, things of that nature. So they can, figure out some anatomical features and what that could have meant in terms of lifestyle. But again, there are definite limits to this. So our best option is the biological species concept, where these populations naturally interbreed and produce fertile offspring. We can look at physical features. Oops, sorry, it's freezing up on me here. We can look at physical features, whether in the fossil record or in species that are around today, but there are limitations to that. So that is how we define species. Now what we want to do is think about, well, how do we know when new species are created? And so the sort of definition that we'll use with the biological species definition or the biological species concept is that when populations have reproductive isolation. What that means is that the populations no longer have gene flow between them. So either they're not mating or they're not producing fertile offspring. So they're sort of a dead end in that regard. So when populations limit gene flow, what that means is they're no longer sharing alleles in offspring. So over time, mutations can accumulate and populations can begin to diverge and become more and more different. So initially you have reproductive isolation. Something is preventing new members of the next generation from being created. And then what that does is it allows mutations to sort of fold in over time, because mutations are inevitable. And that leads to this sort of creeping change in populations that makes them more and more different from one another until you get to sort of morphologically distinct species. They start to look different from one another. So step one is reproductive isolation. If there's reproductive isolation, then by definition we've got speciation. And then over time, as differences accumulate, it's sort of easier to spot them as being members of different species. So that's what we're starting with in this first video. So our different definitions of species, and then the notion that reproductive isolation